Biology students, hey, quick video here. This is your lesson for today. We are going to learn how to do a monohybrid cross where we predict the probabilities between two parents, all right? Things that we're going to incorporate that we have previously talked about. Terms like heterozygous, homozygous, dominant, recessive, genotype, phenotype, and we're going to use probabilities. So a lot of things that we have covered are going to be used in this lesson. Once I go through this lesson and show you a few examples, I have a worksheet on the Google Classroom that I need you to open up, and on a separate sheet of paper, you don't have to print that out, you can just use it as a reference sheet. On a separate sheet of paper, work these problems out like I am working out for you here, okay? Hope you guys are staying warm, staying safe. Let's get cracking, okay? All right, so I'm gonna turn this thing down, okay? I'm gonna rotate it so it's a little bit right side up for you. All right, I hope you guys can see that. So I'm going to show you how we do Punnett Squares. What I'm going to do first is I'm going to show you a cross between a homozygous dominant tall, and we're going to use the letter T, and we are going to cross that with a homozygous recessive short, and we're going to remind ourselves that we'll use a little t here, okay? And so what we're going to do is, first thing you want to be able to do is when you get one of these monohybrid crosses, and the reason why we know it's monohybrid is because this is just one trait, how tall or how short a plant is, so plant height. And so what you want to make sure is you want to make sure that you know what the parents here look like. And so that's the first thing you want to write down. So homozygous dominant is going to look like two big T's, and we're going to cross that with a homozygous recessive. So that's going to be two little t's. So this is the first thing you want to make sure you get right. All right. So the next thing we're going to do is we're going to set up what is called a Punnett square. All right. And a Punnett square is going to be something that looks like this. All right. And what we're going to do is we're going to put one parent here, and we're going to separate these two alleles at the heads of these two columns, and then we're going to take this parent and we're going to separate that parent over down the side here like this. So we got one parent segregated here, and we got another parent with the alleles segregated. And then what we're going to do is we're going to fill these boxes in by pushing these T's down and pushing these T's across. And so what we're going to do is big T and then little t, capital T, lowercase t, and then we got one that looks like this, and we got one that looks like this. So what we've done is we've put a big T in each one of these and a little t in each one of these going across. And so between two parents that are this, homozygous dominant tall, homozygous recessive short, this is the probability of our offspring. And so what we want to do is we want to read off two things here from this. We want to read off the genotypic ratio, and we want to read off the phenotypic ratio. Oop, not ration. Okay? Now if you'll remember, genotypic is describing it like this, or looking at just the letters. And so the way I would see this is I would describe this as heterozygous, or big T, little t. And so what I'm going to do is I'm just going to say big T, little t, and I know that that is heterozygous. And then I want to do my probability, and probability is how many times do you see it possibly observed out of the total. So the total is 4, and so in this case 4 out of 4. So the fraction would be 4 out of 4, decimal 1, percentage 100%. I'm going to give all three of those to you so you guys can get used to seeing how all three of these look. So heterozygous, between these two, all of them would be heterozygous. Right? Phenotype is just the physical description. This is where we're thinking tall or short, tall or short. So if I look at this, remember the principle of dominant recessive means that in this scenario we would only see the dominant, this recessive will be hidden. So this would be tall, 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 tall. These would all be tall. So I would write down here tall, and that would be 4 out of 4. decimal would be 1, and percentage would be 100%. So there's no chance of any other type of genotype, and there's no chance of any other type of phenotype. Got that? This is basically how you set up a Punnett square. I'll do one more for you here. Okay. Let's do this. Let's do a heterozygous tall. We'll just use the same trait. We're going to cross that with another heterozygous tall. All right? And so we're going to use the same letters we had up here. Okay, same letters. We're not going to change T. And so heterozygous means one parent is this. And we're going to cross that with another parent that is also heterozygous. So then we're going to set up a Punnett square. All right, we're going to do like we did up here. We're going to take this parent and we're going to segregate, principal segregation. We're going to put one T here and another T here. And then we're going to separate this one and we're going to put it down the sides. We're going to put a T here and a T here. And some students are like, hey Foster, does it matter how you do this? If you put a little T here or a big T here? It doesn't really, not on a monohybrid, all right? But when they get bigger, there's a certain way we're going to do it just so everybody's can kind of look the same. So now what we're going to do is we're going to drop these T's down. We're going to push these T's across. So then we have two big T's. And then in this box we'll have a big T and a little t. And then in this box we'll have a capital and a lowercase. And then in this last box we'll have two little t's. And so now what we're going to do is we're going to read the genotypic ratio. And I'm just going to abbreviate that G because I don't want to write the whole thing out again. You're welcome to do this. I'll understand what you mean if you write a G on me here. So the genotypic ratio is I just want to look at the kind of alleles that are collected here. All right, so it looks like I got homozygous dominant, so I got two big T's. I'm going to go ahead and remind myself what that's called, homozygous dominant, and it looks like I got a 1 out of 4 chance. So 1 out of 4, 0.25 decimal, 25%. All right, I'm just giving all this to you just so you can get used to seeing that. So that one's done, all right, and then I'm going to go to this next one, and this is heterozygous. I'm going to remind myself that that is heterozygous. I'm abbreviating that. You're welcome to do that too. And it looks like I got 2 chances out of 4. I'm going to write 2 out of 4. You can certainly write 1/2 if you understand that. And that's 0.5, and that is 50%. So these two are done now. And I got one left, and it's this one. 2 little t's, and that would be homozygous recessive. And that would also be 1 out of 4, 0.25, 25%. All right, I've read all the possibilities off here, and sometimes you will catch them referring to this as a 1, 2 to 1. 1, 2 to 1, and that's where these numbers come from. They come from the 1 over 4, 2 over 4, and 1 over 4. And so in case you ever see this, this is how they're referring to this 1, 2, 1, this chance of observance out of 4. Okay? So we're going to do phenotype now. And I'm going to scoot this up. Maybe I'll just point it up a little here. Now we're going to do phenotype. And keep in mind, we are just thinking of, is it tall or is it short? So 2 big T's got to be tall. One of each, we're only going to see the tall. And we're only going to see the tall, and then we got a short. So what I'm thinking is, for tall, we're going to have 3 out of 4, 0.75, 75% chance. Between 2 heterozygous parents, I would expect a 75% chance of being tall. And then we're going to go short, 1 out of 4, just that one, 0.25, 25%. And so now we've got lots of information here. Between 2 heterozygous parents, you can see what kind of genotypes are possible and how those probabilities, 25% of the time you'd expect this, 50, 25, and then tall and short. All right? So that's just kind of the basics. Now what I want to do is I want to kind of go through an actual, a little bit different example for you here. If you would be so kind, I shall do that. Let's see here. Let's do something like this. Let's go with, all right, let's do one involving blonde hair. Okay? And let's say for blonde hair, let's say we want to know what's the chance. Let me make sure that that is showing up. Yeah, good. What's the chance of getting a child with blonde hair from parents who are heterozygous blondes? And blonde hair is dominant in this case. We'll use big B. So with blonde hair being dominant, what is the chance of a child with blonde hair from parents who are heterozygous blondes? So here's what we would do. First thing is make sure you get the parents right. It says they are both heterozygous blondes. So we would do this crossed with this. And then we would set up our Punnett square. I'm going to do this pretty fast. We would split one parent up. And we put one parent here, see? And we split this one and put it here. Alright. And now we are going to do the genotypic ratios. Oh, let's fill this in first. Big B, big B, big B, little b, big B, little b, little b, little b. And then genotypics. Looks like we got a big B, big B. We got a 1 out of 4. I'm just going to go with fractions here and percentages. I'm going to leave the decimals out for now. Looks like we got big B, little b. We got 2 out of 4. That's a 50%. And then little b, we got 1 out of 4, 25%. All right, phenotype. So blonde hair is dominant. All right, it doesn't say anything about what the alternative is, so we just call that not blonde. All right, so blonde hair is dominant, so it looks like we got blonde, blonde, blonde. So blonde, 3 out of 4, 75%. And then we got one chance of not blonde, I'll just say, or non, or whatever. And that's 1/4, 25%. Now before we're done, this is a lot of good work we've got here. And before we're done, we want to answer the question, what's the chance of a child with blonde hair? And so when you do all this, you can see you have an answer right here, and so you just want to circle that. All right? You just want to circle it and say you're good to go. Okay? So, several examples for you to work out. Now, what I have for you on the Google Class is I have a I have a worksheet for you to do. It's got several of these for you to work through, okay? I'll be available 10 to 12. You can email me if it is necessary for you to do Zoom. Just let me know and just get on the Zoom link that we use for our regular class time, and we can get on there and I can help you work through a few things, okay? But you can just email me questions and say, hey Foster, I worked through this one, I was getting this kind of result, does that sound right? We can certainly do that, okay? Hey, I hope you guys stay safe and stay warm and all that stuff, and I hope you get some handle on how to do monohybrid crosses, all right? All right, it's Foster. Take care.